All right. All right. Well, again, this is my first talk. And again, I wanted to say what an unmitigated pleasure it is to be here and see you all. And it's wonderful to be invited, and it's wonderful to be at events like this. It really reaffirms my faith in humanity, especially younger humanity. You know, you younger generation are some of the most thoughtful and intelligent and kind and compassionate people that it's ever been my privilege to know. And I really think that's one of the rewards I find so much in teaching, and because it gives me a chance to interface and get to know the next generation, the folks that are going to get us out of this mess that we've gotten ourselves into. If anybody can, you're the people. So, good luck with that. Because we old people, we're going to be drooling in our gruel pretty soon, and we won't have to worry about it. But, as long as there's somebody to change our diapers, so hopefully this interchange that we're going to have helps with that. So the topic of this talk is neuroscience and spirituality, if you haven't already been paying attention. So let's get into it. The human brain has sometimes been called the 3-pound universe. And a famous quote is, "The universe exists for us only insofar as it exists in our brain. The brain is the 3-pound universe. Each of us lives within this universe, the prison of his or her, of course, own brain." And I've always liked this diagram from an early anatomical text from 1543. For one thing, it's always good to shock my students to look at this. This is made from an actual anatomical model from a cadaver. I've always enjoyed the expression of the gentleman who donated his brain to science for study early on. So I guess the point of this partly is that the brain is probably one of the most complex and highly organized objects in the known universe. And if you've studied systems biology, you have a notion that complexity and high degree of organization is one of the chief characteristics that characterizes biology. This is one of the things that separates the living from the non-living. As matter self-organizes itself into complex structures, you get emergent properties such as living systems. And as living systems become ever more complex, you get structures like the human brain that display further emergent properties, in this case, the emergence of consciousness. Well, is the brain the seat of the soul? We don't really know. We're comfortable with the notion that pretty much everything that goes on in consciousness and our own subjective experience of it, our thoughts, our perceptions, dreams, memories, imaginings, aspirations, and trepidations, all that makes us human are somehow intimately linked to that few 100 grams of spongy, gelatinous tissue that resides inside our skulls. If you look at, it wasn't always so, if you look historically at evolutions of the sort of the intellectual evolution of the notions of the brain and what it does, it's rather surprising because early on it was not necessarily a foregone conclusion that the soul or the mind or consciousness was really connected to the brain at all. Aristotle was really the first to make that connection. which seems to me late in human history that this notion that the brain has something to do with consciousness should emerge. But he thought that the brain was a place where all the spirits came together in common, and that's really where our term common sense comes from. So we have more or less gone with that intuition. We think that the brain is not separable, that it really is somehow inseparably linked with who we are and who we think we are. But in history, one of the conundrums that we face when we're trying to think about these things, to think about our brains, to think about consciousness, is that we're, in a sense, we're the brain trying to think about the brain. We're trying to step out of the box and understand it. And this is very difficult. The conundrum that we face is very similar to a fish trying to understand water. How do you step outside the box of your own consciousness to get the perspective on what consciousness is? This is a bit of a dilemma. And that's perhaps why consciousness is the last frontier in a sense of understanding our relationship to the universe, because we're in the box and we're trying to understand the box. Well, Descartes, of course, came along, and he's the one of I think, therefore I am fame, and the person, the philosopher who originated the Cartesian dualism, the idea of the separation of the mind and nature, and the difference between what goes on inside and what goes on outside, and all of these profound metaphysical questions around existence and being, which have really in some ways not been helpful in the evolution of Western philosophy, because it's introduced a lot of assumptions into that relationship that don't really hold. The notion that we are somehow separated from nature, that we're somehow not part of nature. And this has plagued Western philosophical understanding ever since. Descartes had an interesting notion that the pineal gland, which is the gland, essentially the third eye chakra, as you probably all know, was somehow the site of consciousness, that it was the organ that regulated consciousness. And it did through via a series of strings. The gland was the interpreter of vision, not the receptor of vision, but the interpreter of vision, or the processor of vision. And then one could respond to visual input through a series of literally strings or pulleys. So his notion was that the body was kind of a puppet. And, the brain, the pineal gland specifically, was inside there pulling the strings. And it's sort of the notion, again, like, is it me, or is it the little homunculus inside of me that's kind of running the machine of the self? And these dualistic notions have persisted, and they're so ingrained in our way of thinking about these things. But eventually the picture became a lot more complex as things evolved. Da Vinci, shown up at the top left, had a notion that he really could be responsible for the first serious notion of an airhead, because his idea was that the brain, the cranial space was basically a place for humors or vapors to move in and out of the body, and that the conscious activity was reflected in the movement of these humors. So he postulated that there wasn't much going on up there at all. It was just a medium for the movement of these sort of miasmic vapors, and that was the activity that generated consciousness. But then, as neuroanatomists even early on started to look at the brain structures, they could see readily that it was a lot more complicated than that, and that the brain was indeed a very complex, complex structure. And so, naturally enough, as they dissected these cadavers, they tried to relate the function of the brain to the structure of the brain, because that's what anatomists do. In a sense, they define the structure, but without the other side of that, the process side, which is sort of the province of the physiologists. You can see these complex structures. The question is, what do they do? So eventually, it emerged some of the specific brain structures that they could isolate. There were attempts to correlate them with specific areas of the brain. In other words, to define structure and function and define the link between them. One of the kind of oddball offshoots of this in the 19th century was phrenology, which was really a pseudoscience. It was the notion that by mapping and feeling the bumps on your head, on your skull, you could relate these bumps to what was going on inside. And the phrenologists would develop these complex maps of the skull, and that was supposed to reflect some inner structure function correlation, and they could feel your head and define the bumps, and then they could say, what you were strong in, what talents you had, and this kind of thing. And they could tell much about your personality. But, it was total bunkum. I mean, there's absolutely no basis for this, at least scientifically, as far as we know. But so, that was a theory that didn't have much longevity. Well, as our tools improved, our picture of the brain and its structure-function relationships improved also. And as new techniques came along, which we now have different brain imaging techniques and this sort of thing, we had better tools to try and understand this structure-function relationship. What is the ultimate question is, what is the relation of the physical substrate of consciousness, which we now postulate is primarily located in the brain, although not entirely, and the process that we understand as consciousness, that we experience as subjective experience, our experience of being in the world. Well, now, building on the work of all these anatomists and physiologists over the last couple hundred years, and neurochemists, we shouldn't neglect that, we have a pretty good map, we think, of what parts of the brain govern what kinds of activity, what kinds of functions. So we can pretty well say, in the auditory cortex processes auditory input, the visual, the occipital lobe part of the brain processes vision, olfaction. So in other words, we can come up with a much better map of where in the brain particular functions that go together that are integrated together to produce subjective experience and sensory experience are localized. And of course, science is an ongoing process. So even now, these things are still being more closely defined and revised. None of this is set in stone. But I think there has been progress. And the picture that has kind of emerged from the anatomical point of view is there really isn't just one brain. There's at least three. And those of you that have taken anatomy or read about this already know this. There is, of course, the midbrain, the hindbrain, and the forebrain. The forebrain at the top being the evolutionarily more recent neocortex, this hypertrophied neocortex, which is kind of unique to human beings and is thought of as the seat of what we identify as human processes, the consciousness, the decision making, the ego, all of this. Well, it is not necessarily correct, But this is the picture that's emerged. The hindbrain covers more of the autonomic functions. It's the housekeeping functions of the brain that go on in the background. We don't have to pay too much attention to that, fortunately. We don't have to remind ourselves to breathe, for example, or remind our heart to beat in most cases, because the forebrain and the autonomic nervous system is just handling all that. And in the meantime, we're busy being conscious and having conscious experience, or being busy being unconscious sometimes. But so this picture that has emerged, the brain brain is made up of various lobes. There's basically, as you saw from the previous diagram, the brain is bilaterally symmetrical. There are two hemispheres, the right and the left, and they pretty much mirror each other in terms of their structure. So if you look at a side view of one of the lobes, then you can more or less define these different lobes, the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. Now, of course, in the actual neuroanatomy lab, they are not nicely colored and differentiated this way. This is an artist's rendition, but you can pretty much see that, you know, there are different lobes of the brain. So one way to look at this, so they've sort of focused on the lobe, and within the structure of the lobe, the neural architecture that mediates these different processes. So if you look at we now have a pretty good map of the functional areas of the cerebrum. I'm going to sit down. It's actually easier, I guess, for me to work it from here, if that's all right. So if we look at the auditory association area, that is pretty much located in the temporal lobe, and it processes complex auditory information. And the auditory cortex, also in the temporal lobe, basically it's the receiver, it's the microphone that detects the input, and then the association area, through complex processing, makes sense of the input. And you see this again and again. The motor association cortex, located in the frontal lobe, coordinates movement. The prefrontal cortex, also in the frontal lobe, controls higher order thinking and emotions. The primary motor cortex in the parietal lobe initiates voluntary actions. This is the part of the parietal lobe that is damaged in conditions like Parkinsonism and other types of motor impairment diseases. The primary somatosensory cortex, again part of the parietal lobe, receives tactile information, in other words touch input, and controls our response to that. The sensory association cortex is distributed between the parietal lobe and the insula, and it processes multiple bodily sensations, but not so much visual input. It promotes touch, smell, taste. In a way, what tactile information and chemically sensed information is processed in the sensory association cortex. And the speech center is, of course, the language center. It's located, one of the language centers. There are actually two, at least, that we know of. In the frontal cortex, the Broca's area, it's sometimes called after the neuroanatomist that discovered it. It governs speech production and articulation, the movement of the mouth, and an enormous amount of the neural architecture in the frontal lobe is devoted to governing the movements of the facial muscles. Some of you have probably seen the little homunculus that's sometimes shown in neuroanatomy books with the with the face, the body image is mapped to the area of the frontal cortex, and the mouth and the muscles governing the mouth is this enormous face, and the rest of the body is kind of an appendage hanging off that. So, the speech center is an enormous component of the complexity of our brain, and is one of the reasons why the human brain is so complex, because, language is one of the things that really sets us apart from the other creatures, even other mammals. Now, other animals do have language, but it's rudimentary compared to ours. And we'll talk a lot more about that tomorrow night. The visual association area is located in the occipital lobe, the back part of the brain, and it's responsible for visual recognition. And the visual cortex, again in the occipital lobe, receives the visual input from the eyes. So again, you see, these areas are associated. You've got the visual cortex receiving the raw data, and then an adjacent area of the brain actually processes that data, in a sense decrypts it, you could think of it, or filters it and makes it make sense to us. So the function of recognition and interpretation is sort of laid on top of that raw input, very much like you see with auditory input. And then another area of the brain, Wernicke's area, is another language area. It's located in the temporal lobe, and it's involved with language comprehension. So Broca's area is involved with speech production and controlling the movements of the mouth and facial muscles needed to produce language. But Wernicke's area is associated with the associative areas of the brain, and it has to do with interpreting these small mouth noises that we can't seem to stop making, that are inherently meaningless, but the brain supplies the meaning, supplies the interpretation. Okay, so we have a pretty good picture of the lobes of the brain and how these functions are divided and mapped onto the brain, and kind of how it all goes together. So, do we understand where the soul is? Have we got any better handle on what the soul is? We have a good definition of what it is, but we don't know what the soul is. And we don't really know what the relationship is between the brain and the physical substrate of the brain and what we subjectively experience as consciousness. But we have some ideas. We have some educated guesses, basically. So there are some key questions that we still don't have the answers to, and this is the frontier of neurophysiology right now, trying to answer some of these questions. Big questions that have troubled people ever since they bothered to ponder these problems. What is consciousness? That's a biggie. We know it exists, at least we think we know it exists, because we all experience it. When we're not asleep, we're pretty much conscious. So we're immersed in consciousness all the time. Subjectively, we know it exists. Does it originate in the brain? Does consciousness originate in the brain? Well, we think so. We can't actually say that for sure, but the assumption is that, the brain is the generator of consciousness. This piece of gelatinous meat inside our skull is able to generate consciousness. It'd be nice if we knew what consciousness was, but that's our assumption. Okay, so what is the self? In our conscious experience, unless we're schizophrenic, for example, or have other types of mental illness, or have different types of impairments, we generally experience our own existence as an integrated self. We have a sense of self, of identity, a sense of I, and so on. But so what is that, and what constructs that, and where does that come from? And where is the self in the brain? Is there a little homunculus in the brain that's kind of like running a steam shovel, pulling these levers and pushing the buttons and making the whole thing go? Or, can you go in and cut out an area of the brain where the self resides, and after you sew the person up, then they don't have any self anymore? Well, actually, in some ways you can do that. Some of the work with neurophysiology shows that you can seriously disrupt a person's sense of self, self-integration, their body image, their notions of location in space and time, their memories and all this. But there's no one spot that we can point to and say, okay, there's the self, it's sitting right there. And I'll just aim a laser right there or whatever and ablate that, and then bye-bye self. It's not that simple. So these are, kind of eternal questions. And we're not going to answer them tonight. I don't think. But we're going to cover some interesting territory, in the process of kind of meandering around this question, if not answering this question. One of the things that's fascinated me, and I don't really think there is a solid answer, is consciousness something that the brain generates? That's the usual assumption. But the alternative assumption is that consciousness is something that permeates the continuum. And the brain is a detector of consciousness, much in the way a radio is a detector of radio waves. And, we're all surrounded by radio waves here all the time, but we're not aware of it until we turn on the radio and start tuning it, and then all the stations start to come in. And I think our experience of psychedelics does open this possibility, because we often have that sense. And, where does all this stuff come from? Does our brain generate it, or does it come in from the outside? All these ayahuasca beings and the others and all that, and of course, a reductionist psychiatrist or neuroanatomist would just say, you've screwed up the wiring of your brain and you're having hallucinations or delusions. But that's not our subjective sense of it. It's much more integrated and coherent than that. It does not, the psychedelic experience does not seem to be, the random noise generated by a diseased or disordered brain. So I think the signal is still, the question is still out. The brain may as much be a detector of consciousness, a detector of the signal, as it is a generator of consciousness. We really don't know. Now, we can talk about different, what you might call neurotechnologies. In the last few decades, in the last couple of decades really, there have been incredible advances in the tools that have come available to us to examine these questions. Whether consciousness is generated by the brain, whether it's detected. We can actually now, using these new neural technologies, we can sort of peek in on the brain while it's doing stuff. We can look at the brain in different states, and we can look at what parts of the brain are activated and so on. So more than ever, we have a window on the sort of neuromechanics of consciousness and subjective experiences. A lot of these neurotechnologies fall under the category that we call neuroimaging. And there are several that are quite important, that are quite useful. One is electroencephalography, which is a way of measuring the discharges of electrical activity across the surface of different lobes of the brain. And you've probably seen pictures of, if you saw National Geographic a while back, of the Tibetan monk hooked up to a net of electrodes. It used to be we could only look at a few channels at a time, but now we can, with processing capabilities being what they are, we can look at hundreds of frequencies at a time and get a fairly more comprehensive picture. Another one is computerized axial tomography, or CAT scans. This is basically an X-ray method for making three-dimensional images of brain regions. Another one that's quite exciting is MRI, or so-called fMRI, which is functional magnetic resonance imaging. It's functional because it gives you it's dynamic. You can use it to map what's going on in brain structures in real time. Another one is PET, positron emission tomography. Another one is SPECT, single photon emission computerized tomography. I'll talk in more detail about each of these in a minute. So we have these neurotechnologies, these brain imaging techniques, and thanks to computers, basically, and technological ingenuity, we have these techniques. But what we also have that are important tools are what we might call altered state technologies. In other words, these are what we can apply. These are the probes that we can apply to the brain to see what happens when we're looking at what's going on in the brain during altered states, or even during so-called customary states. So altered state technologies include obviously things like psychedelics, shamanism, meditation, and prayer. And we can use these tools in conjunction with neuroimaging technologies to get an idea of what is going on in these different states. So this is electroencephalography. This is just shows, it's basically a way to map electrical activity in populations of neurons. As the brain functions, as it thinks, or whatever it is that it does, there are waves of electricity that move across the cortex, and they can often be correlated with conscious experience. So this is a way to look at these waves and what areas of the brain become activated on ayahuasca, say, what are the frequencies that are activated, how does that differ from, say, sleep or dreams, and that sort of thing. So EEG gives us the tools to measure the electrical activity in the brain. CAT, or CT, computerized tomography, is not used so much anymore for neuroanatomy because the resolution is very low, but it's basically an X-ray technique. That's another reason it's not used, because X-ray, X-radiation is harmful. But it's a way to, in the living brain, take a slice, a thin slice of a section of the brain and image that to a screen. And by superimposing these images, you can build up a three-dimensional picture. But it's not, the resolution is rather poor. Interestingly, one of the other applications of CT technology is airline security. When you put your baggage through one of those scanners at the airport, that's what they're doing. They're giving it a CT scan, which is maybe why that's not very effective either. Poor resolution. They can't see the pocket knife that you're smuggling through. Or perhaps the cigarette lighter in my case. Anyway, fMRI is much more, gives much better resolution now, and it doesn't involve radiation, as such. So it's preferred now to CT technology. MRI stands for magnetic resonance imaging. And what it is it's based on the absorption and emission of radio waves by protons in water inside the cells. So when you're put into the tunnel, you're exposed to a very powerful electromagnetic field, and this excites the protons, and they give off a resonance that you can detect by another magnetic detector. It doesn't hurt the tissue. Apparently it has no effect as far as we know. And the fMRI reiteration of this or advance of this, basically MRI gives you a picture of static structures, still very useful, but fMRI will actually give you a dynamic picture of what's going on. So very useful technique. This particular movie shows the activation of specific brain areas in a depressed patient after several weeks of treatment with antidepressant medications. So that's what the little red dots are there. Don't ask me what the interpretation of this is. I just ripped this off the internet. So nice picture though, yeah? Okay, and positron emission tomography is another dynamic neuroimaging technique. Dynamic being that it gives you, a movie. It gives you a real-time picture of what's going on. And the way that it works is you put into the brain a positron-emitting molecule, usually a glucose molecule. You administer it intravenously, it goes into the brain, and as it decays, you get that antimatter-matter interaction. This is at very low levels, but the glucose, since glucose is the fuel of the brain, that is what it eats, that's what it needs to function. So there's a correlation between the areas of the brain where the glucose is being metabolized most rapidly with brain activity. So if you can say, okay, you use the ligand that is used or the drug that is used is fluorodeoxyglucose. So it's a glucose analog. And if it goes mostly to, say, the frontal cortex when you're thinking, you can map this using PET, and you can say, well, most of the activity is going on in this area and that area of the brain. So that is also very useful. SPECT is another similar technique. I couldn't find any animation for this one, but it's another way to measure blood flow. It's another way to measure, it's another dynamic imaging technology for measuring blood flow. Instead of using glucose, you can actually inject a labeled, a radioisotope labeled drug, pharmaceutical, into the brain. So if you want to, say, image the serotonin transporter, for example, you can label a drug like Prozac, for example, a selective serotonin uptake inhibitor, with the appropriate isotope and inject that and see how the brain metabolizes that. So and then the radiation that is emitted for SPECT is gamma rays. So then the SPECT detector is essentially a gamma ray detector. These techniques are marvelous. They give us the tools to watch what the brain is doing when the brain is doing whatever it is that it does. But then it becomes even more useful when we combine it with altered state technologies, or what you also might call technologies of the sacred. The neuroimaging technologies give us the windows, but the altered state technologies, these very ancient techniques, give us a way to put the brain into specific states, and then we can sort of spy on it and see what is going on at the neuroanatomical level, at the physiological level, when we're having a psychedelic experience, for example, or a meditative experience, or a mystical experience, or sleeping, or waking, or, you know, when we're looking through a catalog and lusting after, you know, that new Mac that I like, or perhaps the BMW that somebody'd like to buy. In fact, these techniques are all being used for marketing and things like that. This is sort of the dark side of neuroimaging, because marketers can actually take focus groups and put them in an fMRI and then show them, you know, Pepsi versus Coca-Cola, for example, and see, you know, what lights up. And this is being done. This is definitely being done. Neural marketing is a whole new area to, you know, get us to. They can figure out what, you know, they can get right into the brain. And so anyway, meditation is obviously one of these altered state technologies. Meditation, yoga are one way to develop altered states without the benefit of drugs. So they're a way to call on our own neurochemistry to induce altered states. So as a way of sort of defining a baseline, meditation is very useful because there's nothing introduced into the system. It's just the brain's doing what it does in a state of calm relaxation. And so you can see, you know, what areas of the brain are activated or not, or suppressed during a state of meditation. Now, you can also extend this to, well, in the case of psychedelics, psychedelics are, in some ways, you can think of them as molecular probes for studying consciousness, for studying the relationship between the brain and the mind, the so-called brain-mind interface. And as you know, if you know basic pharmacology, the way drugs work usually is they bind to various receptors in the brain, various neurotransmitter receptors. In the case of psychedelics, they interact with serotonin receptors. And this picture on the right there is a diagram of the LSD molecule nesting into the receptor site of the serotonin receptor, in this case the 5-HT2A receptors. So this is very useful because we can define relationships between the molecular configuration or structure of a psychedelic molecule and the kinds of subjective states or conscious states that it produces. Now, prayer is another useful altered state technology that can be studied using these techniques. Prayer, meditation usually focuses on an inner state. It's really about the communion with the self. And it differs from prayer in that prayer usually involves communication or communion with something outside the self, or something that is understood and thought and perceived to be outside the self, whether it be, you know, whatever you wish to call that thing. But it has somewhat difference. It is a somewhat different state of mind. But in both prayer and meditation, ego consciousness is suppressed, so they have that in common, and you can watch what's happening. Now, this gentleman here is a neuroscientist at UCSD, runs the Center for Brain and Cognition laboratory, and he's written several popular books. The one that I'm most familiar with is a book called Phantoms in the Brain. And he has written some very interesting popular books that try and relate the data that's coming out of the neuroscience lab and out of these brain imaging studies with our own subjective experience of self and mind. And he talks about qualia. Qualia, subjective experiences involve a sense of quality. We have, our subjective experience is that we're pretty much located in space and time. We have a sense of separation between ourselves and the external environment. We have an integrated notion of who we are and where we are, and as we move down the stream of time. And so this is one of the questions that he's trying to answer. We agree pretty much provisionally that consciousness is located in the brain, but how does this neural machinery result in experience, what we experience as consciousness? Where is it located? And what's the relationship between qualia, our qualitative experience of being, subjective being, and the neurophysiological processes that are going on in the background that we're not really aware of except through subjective experience, unless we happen to be in an MRI machine, then we can see other aspects of that. So how does the brain put it all together? How does it actually create this experience of qualia? And we generally experience ourself as one thing. We don't think of ourselves in most of the time as many selves. But one theory that he's come up with that I think is very appealing, because it does have explanatory power, is that there are actually many selves, and each self is mediated by a different part of the brain. And our experience of being and subjective awareness is a composite of this that is woven together and integrated by yet other parts of the brain. So each part of the brain processes different aspects of our experience of subjective awareness, subjective being, and then somehow in the end, it's integrated together so that the brain creates a simulation of subjective experience of our being in the world. This is what some people have suggested. Well, this is the hallucination that we have created, that we've all agreed to call consensus reality, and we proceed to live there. And it doesn't really resemble reality, but we'll get to that. So one of the things that comes out of Ramachandran's work that I'm impressed by is how little of the brain is needed to synthesize reality. Most of what the brain is doing, it's all going on in the background. You're never even aware of it. You can think of it as housekeeping functions in a sense. But the part that maintains the illusion of selfhood is mediated by only a few parts of the neuroanatomy. So he talks about the 7 cells. He postulates 7 cells, and there is the embodied self, the part that maintains our image, our body image, and that is localized in the parietal lobes and projects to the frontal lobes. There's the passionate self, the part that mediates emotions and personality, and that's pretty much localized in the temporal lobes or the amygdala. There's the executive self, the part that does things, that acts on input, makes decisions and takes actions. And that part is pretty much mediated by the anterior cingulate gyrus and projections to the motor cortex. There's the mnemonic self, the self of memory, the self that creates the personal story that we tell ourselves that places us in space and time. And that is localized to the hippocampus, the amygdala, and the temporal lobes. There is the unified self, the one that fills in the gaps in the picture, the one that, if necessary, through confabulation, through basically making stuff up, is involved in weaving all this kind of other input together into a coherent experience of self and being, primarily located in the amygdala and the anterior cingulate gyrus. There's the vigilant self, the one that is involved with perception and tracking, the one that monitors the environment and responds to it, and that is located also in the anterior cingulate gyrus, the thalamic nuclei, and the brainstem. And finally, there is the conceptual and social self, the I function, the ego function, the one that we present to the world, to other people in our social interactions, and that is mediated and controlled by the cerebral hemispheres. And there's no real hierarchy implied here. All of these cells have to work together to synthesize the hallucination that is reality. So the reality hallucination. Reality is a crutch for people who can't cope with drugs. It's really true if you think about it. If you think about how our brain apprehends reality, we integrate the input from all of these different cells, and essentially we construct a model of reality. And then we live in that model. And the model is a provisional model. It's imperfect. The reality itself is unknowable. We never perceive the real world. Physics, quantum physics, and other types of data tell us that tables are not solid, that things are mostly empty space, that it's electrons buzzing around. We don't experience the world that way in most of the time, except sometimes we do when we take psychedelics, but most of the time things seem pretty solid and pretty located in space and time. So what we construct from the unknowable real world is a model world that we live in, and that's based on the input from the real world and the output to our interpretation of it. This is what the brain does. It synthesizes a story and it tells us this story. And our experience of the real world is filtered through this sensory neural interface. And that's really what the brain is doing. So this is how we experience it. And I remember what I wanted to say. We know, or at least we think we know, that there's enough correspondence between this model that we create in our heads and live in and the real world that it has survival value. There's enough of them mapping functions that we don't go around walking off cliffs or stepping in front of buses and this kind of thing, because that doesn't have much survival value. So we know there's some correspondence, but beyond that we really can't say a whole lot, and we can change the reality hallucination rather easily. We can take drugs, for example, or we can engage in other types of altered state technologies. Well, the unknowable real world doesn't change, nothing out there changes, but the model can change quite radically. And that's because we've changed the input data. We've applied a different filter, for example. And by doing that, we've changed the output, our response to the information we're getting. It's not so crazy. Michael was telling me, for example, the platypus has neuroelectrical detectors in its snout, and that's how it locates its prey. So the platypus can see electricity. Insects can see in the ultraviolet. And all the patterns of flowers and so on that you see for pollination, if you look at them in the ultraviolet, they look very different. They're actually targets for landing on the flower. Imagine how different our perception of the world would be if we could see in the ultraviolet, or if we could see electricity, or if we could perceive radio waves, for example. All these things are out there. We just don't see them. They just do not impinge on our nervous system, but that doesn't mean the nervous system can't detect them. So then we can take a psychedelic. We can essentially replace our own serotonin neurotransmitter with the next entheogenesis neurotransmitter, like DMT, which is very close to serotonin. And, all of a sudden the picture changes. Oh my God, reality is structured very differently. It's not what we thought it was. So this is the reality hallucination. We can change the filter, and you change the nature of our model of the world. You don't change the real world, presumably, although one could argue this point, but generally not. But we certainly change our perception of the world. Okay, so one of the questions that inevitably comes up here, and that has been, the subject of some curiosity, is what about God? Where is God? Is God a function of this reality hallucination that we've created, or is God, a part of the brain, or and what neural structures mediate our perception of God? Because we have subjective experience often of deity, or of at least beings outside of ourselves that we like to think are much wiser and all-knowing and so on than we are. So what is the what's the neural basis of the God experience? Well, if you happen to be a hard-nosed reductionist like Bertrand Russell, who I'm sure never took a psychedelic in his life, his take on it is, well, there's no difference between someone who eats too little and sees angels and someone who drinks too much and sees snakes. So that was his take on it. But, he was a notoriously, sort of hard-nosed guy, very reductionist, and didn't, have much to do with all this mystical claptrap, But now we can actually use our tools that we have to study this process and get a better handle on it. For instance, this gentleman here, Andy Newberg, has been using SPECT neuroimaging technology to explore brain states and mystical experiences. And so he's worked with Tibetan monks, among other groups who are very experienced meditators. And he has found that in the image above, for example, during a state of deep meditation, the frontal lobes that are involved in concentration and attention are highly activated. They're much more active than in normal consciousness. Sorry, Tim, but this is what the science says. Or this may be outdated, I don't know. And the image below shows that in the parietal lobes, which mediate our sense of orientation in space of time, our notion that we're localized rather than sort of at sea in some oceanic field of bliss, the suppression of those functions they're basically suppressed. So in a state of deep meditation, we can have the feeling of, cosmic kind of floating in the, somewhere, but not localized. And so, this may lead to this sense of timelessness and spacelessness that are associated with this. Another investigator has looked at what's called the Unio Mystica in a group of Carmelite nuns in Montreal. They are a sect of nuns, Catholic nuns, that basically don't do much except try to achieve this state of profound mystical bliss. Some of them spend all of their lives in states of deep meditation and retreat and all this kind of stuff in order to have this God experience. And so, this investigator, Beauregard in Montreal, managed to persuade some of these nuns to, okay, let me shove you in an fMRI machine while you're having this mystical experience, and which might disrupt it a little bit. But again, when he did that, he found that that the experience involved activation of areas in the temporal lobes, the areas of the brain that involve emotional processing, the sense of passion, those sorts of things, and again, the spatial representation of the self. Some people have said, well, they didn't really have a real Neo Mystica because they only have those maybe once a lifetime, and they were faking it. And maybe so. Maybe this is a legitimate criticism of the work. So some people are content to simply observe the brain in subjects, in patients, during these different types of altered states. But this gentleman at Laurentian University is more of a hands-on type, and he got interested in trying to create these different brain states using some other kind of technology, and he built a machine called the God machine, which he claims can induce a mystical experience, and it uses transcranial magnetic stimulation in order to do that. And it's his theory that many of the so-called mystical experiences that people undergo are due to magnetic stimulation of these different intracranial stimulation. And so people have experiences of God, or experiences of the presence of supernatural beings, or the presence of aliens, demons, succubi, ghosts. All this stuff are basically activations of particular parts of the brain, particularly the limbic system. So he developed this God machine, which is basically an electromagnetic device that applies a transcranial low energy magnetic field across the hemispheres in a patient who's put into an isolation chamber. And he claims that this essentially suppresses left hemisphere functions temporarily and allows the right hemisphere, more integrative, more, less language-oriented, less focused and analytical part of the right side of the brain, in other words, to come to the fore. And it disrupts our sense of localization in space and time. It amplifies emotion, much as the left hemisphere helps maintain a sense of self. The right hemisphere kind of has the opposite effect, the so-called subdominant hemisphere. And the reason that he developed the God machine was he originally got very interested in a phenomenon called temporal lobe epilepsy. Now, neurologists have known for years that a thing called temporal lobe epilepsy, where the lesion is located in that part of the brain, doesn't lead to convulsions in the sense that we normally think of epilepsy, or writhing around, or whatever it leads to. When there's a spontaneous electrical discharge in the temporal lobes, it can activate parts of the limbic system that mediate feelings of everything from feelings of transcendence to feelings of terror to feelings of ecstasy. And there's pretty good evidence now that spontaneous mystical states and mystical experiences may be involved with temporal lobe epilepsy. There seem to be two types, the transcendent type and well, let me see. Yeah, the transcendent type and the terrifying type. And the people who suffer from temporal lobe epilepsy also are often subject to spontaneous rages. They can't control their temperature, their tempers, but they may also have spontaneous experiences of mystical insight and philosophical insight, and so on. So this temporal lobe personality is actually pretty well defined in psychiatry. They often have feelings of a divine presence and of being in direct communication with God. They often see cosmic significance in everything, even in trivial events. They are often obsessively preoccupied with theological and philosophical issues. They have an inflated sense of self-importance. They're egocentric and argumentative. They often practice hypergraphia. They keep elaborate diaries, or they write long screeds on mystical and metaphysical topics. They have transient amnesia, or conversely, they recall minute details of events experienced or books read decades previously. And when I show this slide at conferences like this, I sometimes wonder, are psychedelic aficionados, are we a self-selected set of people afflicted with temporal lobe epilepsy? It would seem that we are, or if not, it certainly would seem that maybe we'd like to be. Typical statements of transcendent temporal lobe epilepsy patients, for example, are, suddenly it was all crystal clear to me. There is no longer any doubt anymore. This is the moment I've been waiting for all my life. Finally, I have true insight into the nature of the cosmos. Well, draw your own conclusions, folks, but, maybe these psychedelics are dangerous. They induce temporal lobe epilepsy of the transcendent type, and sometimes of the terrifying type. But, we're in good company, or bad, as the case may be. These are some people, figures from history that are thought to have had possibly temporal lobe epilepsy because they had spontaneous mystical experiences, and they were also known for having terrible tempers and flying off into outrageous rages all the time. They had problems with self-control. So, this is very interesting, this temporal lobe epilepsy angle, and it needs to be looked at a little bit more closely. Now, another approach to, the God experience, the subjective God experience, as we know, is, of course, shamanism. And shamanism and neurotheology go right together. And to this audience, I don't have to explain this. Shamanism is, of course, the archaic techniques of ecstasy, another way to induce these altered states, and another way to induce essentially contact with the mysterium tremendum, or God, or entities outside the self that are superior beings. And shamanism is really the oldest form of religious practice, and over all the world you see common elements, common techniques, and practices, and understanding of what shamanism is. That whether drugs are involved or not, and drugs are almost always involved, but there is shamanism does happen without drugs. Other techniques are used to achieve ecstasy, but the themes of soul travel, the themes of contact with the spirit world, death, rebirth, and healing really are common elements that you find in virtually all shamanic traditions. And a scholar of shamanism, Dr. Michael Winkelman at the Arizona State University, has written an interesting book called Shamanism, the Neuroecology of Consciousness and Healing. And he claims that the universals of shamanism are related to basic brain functions. The shaman's experiences and practices around the world have fundamental similarities because they reflect innate brain processes. And this makes a certain amount of sense. We all have the same basic brain structures the world over. And so activated, in similar ways, it makes sense that we would have similar experiences no matter what our cultural background is. And these brain processes include the synchronization and the integration of neuroelectrical brainwave activity between the limbic system, there's that limbic system cropping up again, and the frontal cortex, the interpretive part of the brain that makes sense out of all this and affords it significance, and also the activation of the brain's feel-good neurotransmitters, especially serotonin and the opioids, the endorphins, the brain's internal opiate system. Well, we can talk about serotonin receptors, of course. How could we overlook them? We've already been talking a lot about them. Serotonin is one of the master neurotransmitters of the brain. It originates, the serotonin neurons originate in the brain stem in a cluster of neurons called the raphe nuclei, and they pretty much arborize all through the brain, especially the limbic system, the frontal cortex, the amygdala, then the areas of the brain that are involved with the mediation of our sense of self. There are many different subtypes of serotonin receptors, many different classes of serotonin receptors. So, in other words, the neurotransmitter is the same, but there are many subtypes of receptors, and so they differ in terms of where they're localized and what kind of functions they mediate. Also worth noting, serotonin is a tryptamine. The chemical name for serotonin is 5-hydroxytryptamine, and you can see it there at the bottom. So, that's the chemical name, and serotonin is kind of the physiological name, the hormone name. So it's a neurotransmitter, and this is a diagram. I guess my mouse can't be used. This is a diagram of a serotonin neuron, and I guess we all pretty much understand the process of neurotransmission since we're preoccupied with psychedelics, but just a brief way to look at it. At the top there in this little schematic or cartoon.
Laser pointer.
Oh, that's good, thank you. I was wondering about that. Oh, boy, now you're in for it, I tell you. We can go through this whole thing in great detail. How am I doing for time? Just go on, okay, all right. It's really not too long. I knew this would happen. But anyway, so this is a diagram of the synapse, right? And the nerve impulse comes down the presynapse, the nerve impulse travels down that and depolarizes the membrane. The serotonin is synthesized from tryptophan. Tryptophan is an essential amino acid, essential meaning we've got to get it from our diet. And by several steps, it's synthesized into serotonin, and it's packaged into little vesicles, little membrane-bound vesicles that they just kind of sit there. But when the nerve impulse passes down, they cause these vesicles to move to the membrane and fuse with it and release its package of serotonin into this synaptic gap here, this synaptic cleft. And then it diffuses across and it binds with a serotonin receptor on the surface of the postsynaptic neuron, and that either causes the postsynaptic neuron to fire or it suppresses the firing. But anyway, something happens because, and this diagram shows various kinds of serotonin receptors on the postsynaptic membrane. Actually, in actuality, there's only one type per neuron, but the binding of serotonin to the postsynaptic receptor causes things to happen. Second messengers, so-called second messengers, changes the conformation of proteins, the so-called G proteins associated with these receptors, and then you get things happening. which like depolarization of the nerve, and sometimes other things like activation of even gene expression and that kind of thing. Now there is in the presynaptic membrane, there's another protein, and you can think of it as a little vacuum cleaner. It actually sucks up the serotonin from the synaptic gap and recycles it, and that is what they call the serotonin transporter. It's not really a receptor, it's a pump. It moves the serotonin back and it recycles it. This is the thing that things like Prozac block. When we talk about a selective serotonin reuptake inhibitor, Prozac is essentially a little monkey wrench that jams this thing so it can't do that, right? So there's more serotonin in the gap, and that over time, has the antidepressant effect. Now, psychedelics, what we call the classical psychedelics, interact with the postsynaptic receptors, the receptors on this membrane, and a particular class out of these 14 or so, especially the 5-HT2A receptors or the 5-HT1A receptors are the ones that psychedelics seem to target. Now, MDMA and empathogens or entactogens work quite differently. If Prozac, they also work on the serotonin transporters. What MDMA does, if Prozac jams the transporter closed, MDMA jams it open, so that all the serotonin essentially leaks out at once. And this is why it depletes serotonin from the presynaptic thing, and then that leads to, all these, at least at high doses, all these subsequent events that some have characterized as neurotoxicity. It does lead to long-term changes in the arborization of these serotonin fibers in certain parts of the brain. But anyway, that is a sort of a quick look at the function of the neurotransmitter at the synapse. And most other neurotransmitters, dopamine, norepinephrine, GABA, so on, are pretty similar. But our preoccupation is with serotonin, so it's good. It's a good one to illustrate that. Now if we didn't already know it through psychedelics, we know that there's an association between serotonin receptors and spirituality. There was a recent PET study that showed that that the serotonin 1A receptors may be related to people who have an inclination toward religious behavior, religious preoccupations, and temperaments, and a genetically low population of serotonin 1A receptors. So that's kind of interesting. If they have the people's inclinations toward religious experience may be genetically or neurochemically mapped to the abundance of serotonin 1A receptors that's in their brain. Well, we know already that psychedelic drugs, the so-called classical hallucinogens, or what I call sometimes true hallucinogens, LSD, psilocybin, and DMT, in other words, the 5-HT1A, 2A interacting drugs rather than the MDMA type drugs, are really the best pharmacological models that we have for looking at transcendent experiences or mystical experiences. So we can, we know that they interact primarily with the 2A receptors, and we can use these things as molecular probes to study mystical experience. We can, for example, do PET studies like this, using, again, fluorodeoxyglucose as the imaging technique, and then give the patient psilocybin look how different at what lights up in the brain. Essentially, the red areas here in the brain are where the most metabolic activity is going on. And this work was done by Franz Vollenweider. Many of you have heard of him. He's one of the Hefter research people. And he has a PET neuroimaging lab in Zurich. And basically he found that the drug stimulated metabolism in the frontal cortex, the temporal lobes, and the anterior cingulate gyrus. These are the areas involved in emotional response and processing and the integration of sensory and visual information. And these are the areas activated in, guess what? Temporal lobe epilepsy, meditation, religious experience, and interestingly enough in schizophrenia as well. And there are, of course, resemblances between all of these altered states and schizophrenia and other types of, things that are characterized usually as mental illness. So now we're beginning to see a picture of the brain areas and the parts of the anatomy and the parts of process that mediate conscious experience and mystical experience. Another way of looking at these molecular probes is the work that our friend Sasha Shulgin has done for many years. He was interested in the issue of structure-activity relationships. In medicinal chemistry, if you have a biologically active molecule, the first thing you do, you want to make theme and variations of that. In other words, how does the activity of the molecule relate to its structure? This is a common approach in any kind of drug development. It's just that Sasha happened to be fascinated with structure-activity relationships for psychedelics. So he investigated this over many years in his own laboratory. He had permission for a while to do this for many years until finally they got too upset by the fact that he had published his results, on the internet and gave our community the tools to do this for themselves. They didn't really like that at all. But anyway, it was too late. The cat was out of the bag, and many of you all know his works, his two big books, PCOL and TCOL, which stands for phenylethylamines I have known and loved, and tryptamines I have known and loved. And indeed he has. What he did, I guess his main contribution was that he synthesized many analogs of known psychedelic molecules, taking a very simple molecule like mescaline, for example, and making analogs of that, because it's these are basically simple molecules and easy to manipulate. So one of the things he did was he moved this methoxy group up here, and he started attaching different things to this position here, this X, where X might be methyl group. And then he came up with the compound DOM, which was known on the street as STP in the 60s. Or if X was an iodine, then it was DOI. And if X was a bromine, it was DOB, and so on. So he made hundreds of analogs and tested them on himself in the good old scientific tradition, carefully, kept good notes, was a good observer, and his major discovery was that minor changes, trivial changes really, in the structure of these molecules could result in extremely radical changes in their activities. Something like mescaline with a simple molecular tweak could be transformed into a mega hallucinogen, a thing that would last, 36 hours, and you'd be absolutely nailed to the floor, awash in a sea of hallucinations. And he could take the same molecule and tweak it a bit more, and it was completely inactive. So he did this with many things and learned a lot about structure-activity relationships for hallucinogens. Now, he worked primarily with the phenethylamines initially, but eventually got around to the tryptamines. And of the tryptamines, the natural one, the one that is so widespread in all of these acacias and really in the plant kingdom, is DMT, dimethyltryptamine. And DMT is in a lot of ways the most interesting of the hallucinogens because of its structural simplicity, its resemblance to serotonin. It's very, very close to that. And it's only 2 steps away from tryptophan. And tryptophan is a universal metabolite. It's an essential amino acid that we have to get from food because we can't make it. And it's one of the 20 amino acids that goes into make proteins. So tryptophan is universally distributed in all living things as far as we know. And here's a molecule that in 2 very simple enzymatic steps mediated by enzymes that are also universally distributed in living things can transform tryptophan into DMT, a hallucinogen. So it's no wonder that it's the most abundant psychedelic in nature because it's so close to primary metabolism. And a rule of thumb is that the complexity of a molecule, a secondary product, is sort of proportional to how close it is to primary metabolism, in other words, to universal metabolism. If it only takes a couple of steps to convert it, then it's going to be popping up everywhere. And indeed it does. We know of at least 200 species of plants that contain DMT. It's also found in animals. It's also found in the human brain and the pineal gland. It's all over the place. It's undoubtedly it's found in thousands of plants. It may in fact be found in all plants if we had sufficiently sensitive techniques to detect it. We might find that it's present at low levels in literally all plants. So among other things, this has got to be a headache for the regulatory authorities, because literally, I mean, are you going to make all plants illegal? That doesn't seem very practical. Not that they wouldn't like to, but I don't see how that's going to work. So it's all over the place. And Sasha's got a really interesting chapter in his book, Tikal, called Tryptamina Botanica, that I recommend you read, and he discusses this. DMT is everywhere. Nature is permeated with DMT. And what's interesting about the acute DMT experience is that it's not that different from the so-called near-death experience. It's synthetic DMT or DMT itself is not orally active unless you potentiate it with an MAO inhibitor. So if you get around that by smoking it, then it elicits a very short-acting, about 10 minutes, very intense psychedelic experience that resembles dying, in fact. Or, well, I mean, I don't know, I've never died, but people postulate that this is like the near-death experience. And Rick Strassman, the neuroscientist who sort of reopened psychedelic research in the early 90s, did his work with DMT, and his speculation was that DMT, produced by the pineal gland, a massive release of DMT at the moment of death may be the basis of the near-death experience. Well, I can probably skip over this part, but we all know that DMT plus a monoamine oxidase inhibitor can be orally potentiated because the MAO inhibitor protects the DMT in the gut from degradation by this enzyme, so then it can cross into the blood-brain barrier in an intact form. And when that happens, instead of a 10-minute roller coaster ride, you can get a 3-hour or 4-hour, much more gentle kind of ride, but much more useful in some ways because you have a chance to kind of grok it and understand what's going on. And the best MAO inhibitors that we've found for this are, of course, the beta-carbolines. They're close chemical cousins of DMT, very closely related structurally, also very widespread in plants, also occurring in animals. So this is trivial chemistry. This is close to primary metabolism. And they're potent monoamine oxidase inhibitors, and they're often found even in the same plant because they're really just a further step down that biosynthetic pathway. Here's an example of a few for the chemists among you. You can see DMT here and serotonin very close, and then these beta-carbolines are all these tricyclic, these three-ring structures like that. And of course, this is the basis of ayahuasca, as we know. The vine being the source of beta-carbolines, and the leaf from Psychotria viridis, usually in the Amazon, being the source of DMT. So this is, you combine these things and you have an orally active form of DMT. Well, interestingly enough, some of you may be aware of Jace Callaway's early paper, and speculated that similar processes may be going on in the brain based on our own endogenously produced DMT and beta-carbolines. If you're interested, I can give you the reference. It was published in Medical Hypotheses around 1988, I think. But his idea, we know that light regulates the production of melatonin, which is another tryptamine, by the pineal gland. And during the waking period, during waking consciousness, melatonin production is suppressed. So this little cartoon here shows what's going on. In the waking phase, you got your tryptophan coming here and being loaded on the conveyor belt and turned to tryptamine, and a little bit of it is dribbling out and being turned to DMT. Most of it's going to serotonin, right? The eyes are open, most is going to serotonin, and you've got the serotonin pathway activated. And down here in the melatonin area in the pineal gland, nothing really is going on, not much is going on. The DMT is coming and it's falling into the hole, into the toilet of MAO, and it's being metabolized, and it's all good. You have waking consciousness. Okay, at night though, when the pineal is activated to produce melatonin, then during sleep and dreams, you have the melatonin cycle activated. So you still got your tryptophan coming along here, going to serotonin, the eyes are closed, got your tryptamine going to DMT here, but the serotonin is not going through the serotonin pathway, it's going to the melatonin pathway, and it's building up, and the melatonin is churning out pinolene, which is an MAO inhibiting beta-carboline, named after the pineal gland, pinolene, and 5-methoxy-DMT. And the toilet is plugged up because MAO is inhibited by the pinolene, so neither the DMT coming through this pathway nor the 5-methoxy-DMT is being metabolized. So it gradually builds up in the plasma, in the bloodstream over time, and you have dreams, you have REM sleep, you have the visionary states of REM dreaming. Reasonable hypothesis, very easily tested. Well, not that easily, but easily enough. It could be done. As far as I know, nobody has tested it, but this is something that should be investigated. Another aspect that we probably should talk about is synesthesia. Synesthesia is a apprehension of one sensory modality by another. For instance, you can hear colors, or you can see music, for example. You can see sounds. It's translating one sense into another. And some people are genetically able to experience synesthesia. They're just born with the ability, and they undergo synesthetic experiences all the time. But for the rest of us, psychedelics are one of the few reliable ways to experience synesthesia. And there's a lot of evidence that synesthetic ability has to do with language and interpretation. In fact, people who are genetically synesthetic often have a very odd relationship to numbers and words and letters and things like that. A synesthete is, I guess, the right word, will say, well, the number 2 is orange and spiky and smells like cinnamon. You know, and they're not kidding, they're not making it up. For them, that's what it is. Or the letter A, you know, is polka dot and squishy. You know, who knows how they experience it, but this is how they experience it. So we know from these studies with synesthetes that translation of one sensory modality into another has a lot to do with the way that meaning is processed and the way that we associate meaning, images with sounds, right? And that's really what language is. It's a synesthetic process, but we don't even think of it that way. It just happens naturally, and it's unconscious. But that's really what it is. I'm standing up here babbling at you, and hopefully you are deriving some meaning from what I'm saying in the form of images. Although at this point, you know, we may all be beyond that. But anyway, that's the hope most of the time. You know, another interesting phenomenon is so-called out-of-body experiences and the near-death experiences that we were talking about. And again, Michael Persinger, the mad scientist at Laurentian University, claims that he can induce, with his God machine, he can induce out-of-body experiences and near-death experiences. And, you know, it's very hard. You're at the point now where does neurophysiology end and where do paranormal experiences, seeing ghosts, and this sort of thing, or temporal lobe epileptic experiences, psychedelics, you know, what is the brain and what is reality, you know, is it real or is it Memorex, you know, it's very hard to separate, you know, neurophysiological processes and this, what we alluded to earlier about how the brain constructs a hallucination, constructs reality, and we proceed to live inside of it. So you get these themes. You know, the notion of alien abduction is very much linked to temporal lobe epilepsy experiences, or the sense of presence, the sense of, you know, someone is standing behind me looking at me, that kind of thing. And you turn around and nobody's there. You know, I mean, I don't know if the rest of you have that. I think we all have had that experience on occasion. Well, that's that temporal lobe acting up in some ways. And what was interesting about Rick Strassman's early experiments with DMT was that at high doses, people kept reporting these completely crazy experiences involving encounters with aliens. And these were not people necessarily who had ever heard of Terence McKenna. You know, these were people who were experienced with hallucinogens, not necessarily with DMT. They took DMT and high doses, high intravenous doses. They came back reporting that, you know, they were in the saucer being operated on. I mean, the classic, you know, UFO abduction experience. And this was very unsettling to Dr. Strassman as well, who was, you know, actually a pretty straight-laced psychiatrist kind of guy, you know, and he was trying to conduct this clinical study, and it was very much about, well, you know, report what colors did you see and this kind of thing. And no, it was like, you know, I was on the bridge of the starship and they were cutting into my brain and, you know, I mean, to the point finally where where Dr. Strassman basically said, I can't, what am I going to tell the grant committee, right? Literally, so finally he actually terminated the study, and he returned the DMT to NIDA, and he just said, this is so nuts, I can't do science, I can't make any sense out of it. And he left academics and went and joined a Zen monastery to think about it for a while. And he did. And he wrote, as he wrote that wonderful book, DMT, The Spirit Molecule, and finally tried to put it all together. But if you've read that book, he freely admits, this just so did not fit into the pattern of a straight-laced, psychiatrist trying to study a psychedelic drug. He said, finally he said, well, what if it's real? What if these really are experiences? Because there was such consistency between the different stories that people would tell. And basically he said, well, if it's real, I don't want nothing to do with it. It's too damn weird for me, And that's what happened. So, and again, these themes come up again and again. The saucer, the tunnel, the disc shape, the lens, the idea that, in out-of-body experiences or drug experiences or dying, you move toward a source of light and you move through a circular structure. And on one level, of course, you can say, well, you're reliving the birth experience, because that also involves moving through a tunnel into the light. And it may be at the end of life you go through another tunnel into some other light. But it's interesting how UFO, near-death experiences, DMT, and all of these things involve moving, through these circular spaces. So what is going on? Well, nobody really knows. What we can say is a few things. We can say there are these similarities between temporal lobe epileptic experiences, near-death experiences, out-of-body experiences, psychedelics, mystical states, prayer, shamanic ecstasy, alien abductions, and induced brain states, like with Persinger's God Machine. All of these seem to involve activation of certain parts of the brain, especially the limbic system, the part that sort of mediates our sense of self and our sense of localization in space and time. The hallucination that we call consensus reality, and the illusion that what we experience as the self is bounded in space and time, is reinforced and maintained by a relatively few, I would say an astonishingly few, pieces of the neural substructures, these parts of the brain, the cingulate cortex, the limbic system, the hypothalamus, the amygdala, and so on. It's basically the limbic system and its subcomponents that are doing this. And when the function of these structures is disrupted through drugs, through deliberately meditative states or different practices or illness, like epilepsy, when this happens, then at that point, finally, we may experience the self as it really is, infinite, unbounded, and immortal. And that's it.
We'll have some questions.
Okay, my apologies. It was only supposed to be an hour.
Okay, first question.
I'm not sure if you've done much research on the dark room retreat space. I've read some information, but I don't know how scientifically based it is, or if it's just speculative. But from what I can understand, people who experience a dark room retreat for maybe 14 days end up at the end with an ayahuasca type experience.
Mm-hmm.
and so there seems to be a cascade of molecules going from melatonin initially, then to pinealine, then to 5-MeO-DMT, and then to DMT. And the cascade itself, besides being hardwired into the body, seems to be the body's natural way of each one precursors and prepares the body for the next one. So if you start off with melatonin being the hormone of darkness and ending up with DMT being the hormone of light, so this movement through this tunnel.
Yeah.
Could you comment on that, what your feelings are from your neurochemistry?
Well, yes. Hello. Yeah. I mean, I don't know. I haven't read extensively about this darkness process, but it makes complete sense in the context of what we've been talking about here. This is exactly right. Essentially, it's the implementation and the validation of Jace Callaway's theory. You know, you spend enough time in total darkness, what happens is there's an activation of this sleep cycle of the pineal that is persistent, and over time you get melatonin production. You get this melatonin part of that cycle activated. Over time you get the buildup of these psychoactive psychedelic tryptamines in the plasma that's quite persistent. And so it's essentially a way of inducing a psychedelic experience, a beta-carboline, DMT, ayahuasca type altered state without taking any drugs, simply by manipulating your own neurochemistry. And in terms of what we've seen here and what we know now, it makes perfect sense that you can do that. There are other approaches to it as well. There are dietary approaches to it. You can take a lot of 5-hydroxytryptophan, for example, in combination with MAO inhibitors, in combination with methyl donors like S-adenosylmethionine methionine and this kind of thing. And theoretically, you could get a buildup of methylated tryptamines in your system that would be quite persistent. Personally, I haven't done this, and I really have no intention of doing it because the drawback of that is that you're altering your neurochemistry in a fairly profound way, and you can take a drug and the drug wears off after a few hours. And if you take this other approach, it may take weeks or months for it to wear off if you decide that you don't like that, or you don't want to be in that state all the time. And by that time, you've already been taken off to the psychiatric hospital and shot up with Thorazine, and you're off babbling in a rubber room someplace, because you've created a situation where you've decided to step out of the serotonin hallucination and into the 5-methoxy-DMT hallucination. So your reality no longer maps to the reality of your peers, as it were, and so they think you've gone loopy, and in a sense you have. So yeah, I have no doubt really that the darkness therapy can lead to this kind of thing, but I think it's more dangerous than drugs, certainly.
There's a large cultural context, because apparently there's a few different cultures that have been practicing this for many, many years.
Yes, yeah.
The Kogi and the Tibetans and
That's right.
Vedic scriptures. So that
Yeah.
seems like
Yeah, Dan just said there's been lots of cultures.
Lots of cultures doing the dark retreat over many years. Yeah.
Yeah, no doubt. And again, it's not that it just depends on what your preferences are. Yeah, again, we have to look to the indigenous people, who know how to do this because they've been doing it for a long time. So they've had opportunities to make all the mistakes. So there's no point in us repeating their mistakes. Same situation with psychedelics. You know, you want to know how to use psychedelics, talk to the people that have been doing it for 20,000 years. You know, why talk to some button-down Harvard psychiatrist? What the hell do they know? You know, not that, again, I mean, I have respect for Timothy Leary, but I think one of the things that, you know, sort of distinguishes this generation from the previous 60s generation, the reason psychedelics in the 60s had so many casualties is that we had no context for the experience, and we had really nobody to explain it to us. I mean, Tim Leary was out there telling everyone to take psychedelics, but he had no more experience with it than anybody else. He was as astonished as everybody. And he was saying, oh, wow, you know, this is really amazing, and yes, it was, and everybody should take it, but he had no real, you know, way to go back, and, you know, and I think that's where this newer generation, this newer psychedelic generation is, you know, by example, able to make a better choice and say, well, you know, let's go talk to the elders. And, you know, by the elders, I mean the indigenous people that really have worked with these things for a long time. Because they've been down this path, and they've made all the mistakes. So we can learn how to avoid some of the biggest pitfalls if we pay attention to how they do it.
Okay, another question here.
Yeah, regarding the evolution of the human cerebral cortex, I know Terence talked about it like, you know, if it was the pancreas of an elephant that had increased in size so dramatically over such an evolutionary space, a short evolutionary space, it would be unheard of and amazing.
Mm-hmm.
This is the most complex organ, this is the most complex piece of space in the universe, as far as we know of.
As far as we know, right.
Is there a sense of wonder in neuroscience at what the fuck happened? How did this come about so quickly?
There's a sense of wonder. Yeah, I mean, I think neuroscience in general is permeated with wonder. You know, and there's a sense of wonder of the subject of their study. I think they're so busy trying to understand the current brain, the modern brain, and its relationship to consciousness. They don't think too much about the evolution of the brain. I think there's a much more awareness of the anomaly of human brain evolution in paleoanthropology and things like that. I mean, there's definitely a lot of questions about that, but it's not the neuroscientists so much, it's anthropologists that are kind of scratching their head and wondering, you know, how did we get there? And there are lots of theories competing for the idea, and actually much of your question is the subject of tomorrow night's talk. And, you know, my own wacky theory and a couple other people's interesting theories about how this came about. So I'll defer talking about that more for right now. But it is definitely one of the things in evolution that's very hard to explain, how this came about in such a short time. That's the thing. The brain literally exploded in size in over the course of 500,000 years, 1 million to 500,000 years. Nobody quite knows why this happened or how it happened. But tomorrow we'll get into all that. Anybody else? We still have another talk, right?
Yeah, up earlier. Okay, here, I'll come through here.
I can't even see anybody.
There's someone shouting.
Has there been any research done into levels of DMT in animal brains compared to our brains, and like specifically research into if there are some animals which have highly organised social groups like whales or dolphins, elephants, cats, if they have higher levels of DMT in their brain than say animals like, I don't know, something similar like worms.
It's a very good question. It's an excellent question, and exactly that kind of thing. To what degree do DMT involved in organizing social structures and so on? Because as Tim was talking about earlier, there's also the oxytocin connection, and we know that serotonin agonists like DMT and other psychedelics will stimulate, cause an acute rise in hormones like adrenocortical tropic hormone, or prolactin, and presumably oxytocin, although I don't know if anyone's actually looked at this yet. So it's a good question, and the answer is, as far as I know, that this work hasn't been done, but it would be a very fascinating thing to do, very interesting. I don't think anybody's done. Does anybody know about that?
Just empiric.
Someone should do it. One thing about big science, it costs money, so you got to come up with the money to do it. But yeah. I'm not hearing
He's
you.
saying, would it be better to be doing research on rat or rat brains rather than human brains?
Would it be better?
Just easier because
Easier.
Oh, it would be easier to do work on.
What about whales? What about dolphins? What about monkeys? Monkeys with different social structures. For that matter, what about insects and other types of hive-type social organizations?
Jaguars.
Jaguars? Aren't they kind of solitary?
They eat Banisteriopsis carpi.
They do?
Yes.
This is known?
Yes.
Well, okay. I have no idea. Maybe so. Maybe so. I mean, that's an interesting thing too, because that goes to the question of how did the first people figure out to combine these plants?
Dreaming.
The plants told them.
That's what the plants say. The plants told them. But maybe the plants are lying. Maybe they actually watch jaguars. Who knows?
Yeah, that's true.
They eat the copy and they rub up against.
Yes.
Well,
Like catnip.
who knows? It's very possible. I mean, nothing would really surprise me, and the notion that animal behavior might have led to some of these discoveries is certainly plausible. I mean, there's another interesting tale from the Amazon that Mark Plotkin talks about. It's a whole other plant complex, but there's a plant. There are many plants actually, but there's a plant in the Amazon that produces a strong insecticide called rotenone, Daris species, and rotenone is a fish poison, and the people use it as a fish poison. They pound the root, and they will throw the pounded root into a backwater stream, into an area where there's not much water flow, and it'll diffuse into the water, and the fish will be poisoned and float to the surface. But it turns out that, what is it? It's tapirs or something like that. I think it's tapirs. They have observed them. They eat the root of this Daris, which contains rotenone, which is toxic to most things, but apparently the tapir can handle it. They eat the root of the thing, and they defecate into the water. And the root is metabolized, it's in the shit, they defecate it into the water, and then they collect the fish. So they figured this out, you know. I mean, talk about a weird, you know, animal-plant relationship, but there it is, you know. So it's not that unreasonable to think that, I mean, we do know, in fact, that some animals will deliberately consume, you know, fermented fruit, for example, to get drunk. I mean, this is well documented. Elephants, you know, love to consume fermented fruit and will get absolutely, literally staggering drunk.
And aggressive.
And aggressive, yeah. You don't want to be too close when this goes on. This is definitely a 5,000 yards through heavy lenses kind of observation, but yeah.
Okay, question here?
Yeah, slightly off the topic. I was just wondering if you could tell me about pamelaine magnesium. There was something about it the other day.
Magnesium pamelaine. Well, I can't really tell you much about it. You know, it was a memory stimulant supposedly in the, that was around during the 60s, and that's about all I know about it. Anybody, what, with respect to what is your question?
That's a good question.
Okay. Try Arrowwood for that one.
Okay, there's another question. I just have to get to the person's name, excuse me. Right.
We've got a pretty good idea of what all the different drugs are doing within the brain, gut to the receptors. Has anyone actually figured out what the hell nitrous is up to? Because, you know, you kind of have your DMT or your acid and that, and nitrous on top of it, and it certainly increases the effect of what you're getting. So has anyone actually figured out what nitrous actually does in order to produce?
Oh, what the effect of nitrous is? Well, actually, I think there are other people in this audience that can answer that better. Is Michael Bach here?
It's the same as in cars, superchargers.
Yeah, but in terms of receptor pharmacology, he said it was a glutamate antagonist, a short-acting glutamate antagonist, glutamate being another neurotransmitter. And that sounds good to me. I'm not really sure. Yeah?
Hey, another question. Where are you? Come over here. You meet me. Because the microphone works much better over here as well.
Yeah, that's good. I can't really see anybody out there.
Okay, yeah.
I feel good.
Could you just speak a little bit on the corpus callosum, that part of the brain, and maybe what's going on there, and just enlighten us a little bit, or me myself?
Well, the corpus callosum is a kind of a neural highway between the two hemispheres of the brain. It's really the way that the two hemispheres talk to each other, and so it integrates, you know, functions between the right and the left brain. But you can sever the corpus callosum, and this is sometimes done in certain types of treatments for certain kinds of epilepsy. They'll actually cut the corpus callosum, and then what results is that there are really two brains that kind of function independently of each other. And you can still function pretty much, but it leads to some very odd things, very odd effects. For instance, with your left brain, the analytical part involved in naming things, you can inject an anesthetic into the brain and put the left brain to sleep, and then you can ask the person to reach into a box and pick up an object, like a common object like a glass or an ashtray or a block or something like that. They can do that, they can feel it, they know what it is, but they are unable to say what it is. They can't verbalize it. So you get this kind of disjunction between different hemisphere localized functions. And, the corpus callosum is, something that you probably want to keep intact in most cases, but sometimes it's necessary for medical reasons. And you can, it is possible to kind of selectively anesthetize one or the other side of the brain and then see what happens. Is that the kind of thing you're talking about?
That's really interesting in the evolution of the brain. That's the next level.
Yeah, tomorrow we'll talk about all that. I don't want to spend too much more time because we have another talk, is that right?
Yes, that's right, yes.
Okay, well,
Shall we have one more question, Dennis? We've got one person here.
yeah, let's do one more question. Your patience is really remarkable.
Okay,
and persistence.
just a little thought on different sections of the brain and the hemispheres. Now, I've only read very briefly on this, but I've read that if someone has a hemispherectomy, in other words, they have one half of their brain removed, that the other half of the brain takes over the functions of the other half. And I've also read that it actually doesn't seem to matter which half of the brain is removed, that either half can overtake that. Now, if that's the case, then how do we have specific areas of the brain that have localized functions then if another part of the brain actually take over that?
You mean, why are there specialized areas?
Well, how does that even work? How does that even work that if we have, from our limited understanding, thoughts that certain parts of the brain are responsible for certain actions, thoughts, or ways of thinking, how is it then that if one part of that is actually physically removed, that another part of the brain can respond and take over that function?
Yeah, I think I get it. Well, for one thing, this picture of the structural organization of the brain and the specialization of different functions in different areas is a picture of the normal brain, a picture of the way neural development usually takes place. So it's based on, kind of the standard picture and not a pathological picture. But I think your question goes back to this question of neuroplasticity. The brain is a lot more adaptive than we have realized. For a long time it was thought that, you got a certain complement of neurons at birth and it was pretty much, that was all you got. And they slowly died off over time and you lost neurons and you lost functions. Well, that's all gone out the window. We know that neurogenesis, as it's called, or the growth of new neurons, does take place, that the brain is incredibly plastic and able to adapt. And so when there is brain injuries and a certain area of the brain has a lesion in it and is not functioning, adjacent areas of the brain, or often the corresponding area, if it's bilaterally symmetrical, the corresponding area in the other hemisphere can take over these functions. So I think this is just a reflection of the fact that the brain can adapt to the situation, and through neurogenesis and that kind of thing, can compensate for injury and take over these functions. So that's the way it works. And now there's a lot of interest in this neuroplasticity. And this relates to a number of different, things, different interests. It reminds me of a story, and kind of an interesting story, which I'll tell you and then I'll stop. But in my own experience, I was at an ayahuasca retreat a few years ago, and there was a young man there who had severe dyslexia as a teenager. And but he didn't have dyslexia. He's a very smart man. And but as a teenager, he had dyslexia, and it had caused him a lot of problems socially. You know, they thought he was stupid, as they often do of dyslexics. It's not that they're stupid; it's that they, you know, have trouble reading and all that. And so he'd had a lot of problems in school and was sort of socially outcast and all that. So partly in response to this, he started taking LSD, and quite often, and at rather high doses. And he told me that one day, you know, he had taken a very high dose of LSD, and in the trip, he was able to visualize the wiring of his brain, and he could see what he called the filing cabinets where his knowledge of language and interpretation were stored, and where the damage was in this damaged part of his brain. And he could visualize another part of his brain that was unused with a similar installation of filing cabinets. And basically, by thinking about it, he was able to visualize the rewiring of his brain and transfer these linguistic functions from this damaged part of the left brain to the undamaged part of his right brain. And after that, he never had dyslexia. And I thought, I mean, I was astonished by this story, you know, and he said, I was able to read after that. I don't have any problem, and I read for fun, and I just don't have a problem. And I thought, that is truly amazing, and that is a testimony to the sort of unexplored therapeutic potential of things like LSD. I mean, it's not just about, you know, treating depression or cancer, anxiety, and all that stuff, but I mean, that's all very good, but an agent that can actually cure dyslexia, I mean, that's some powerful medicine, if you ask me. And, you know, I think you can do it with these things. Okay, thank you all for listening.
Doing it are so strong that it often pulls others away from their own art.
On your marks. Get set.
Go!
Whoa, push it over!